Okay. Hello, everyone. I think it is time to get started. Good morning, good afternoon, and good evening for some of you. And welcome to our webinar series, New Tools of the Trade in Life Sciences, Technology that Enables Breakthroughs. And today we will learn about MicDrop, a platform for large-scale in vivo CRISPR screens. My name is Renate Weitzbauer. I'm the lead editor of the webinar series here at Bioprotocol, which is an open access online journal whose mission is to make research more transparent and reproducible. Before we begin with the webinar, I'd like to make a couple of really quick announcements. If you have a question, you can post them in the Q&A section or in the chat section throughout the talk, and Saba will address them at the end. We will also record this webinar and post it on our website later. So if you want to revisit it, you can do so later. All right. It is now my very distinct pleasure to introduce today's moderator, Zach Brandt, who is a postdoctoral fellow in Randy Patterson's lab at the University of Utah. Zach received his PhD in cell and developmental biology from the Medical College of Wisconsin in the lab of Brian Link, where he studied the role of hyposignaling pathway in the development and disease using zebrafish as a model system. Since joining the Patterson lab, he has continued to leverage the advantages of zebrafish, using them to model rare or undiagnosed diseases with the goal of developing screening platforms capable of identifying potential therapeutics for patients with these conditions. And additionally to that, Zach has also been using the MicDrop platform to perform high-throughput genetic screens in zebrafish to identify novel regulators of touch sensation. And with that, Zach, thank you so much for being here and moderating today's session, and the floor is all yours.
Thank you. I have the pleasure of introducing my colleague and coworker, Saba Parvez, who will be presenting the technology that he's developed in Randy's lab here at the University of Utah. And I will make it somewhat brief because I know Saba's probably mostly interested in talking about the technology, but just to let you know a little bit more about him, Saba received his PhD in chemistry and chemical biology at Cornell University in the lab of Imon A. And as you will see, Saba's always been interested in technique and tool development. And during his time as an HHMI International Predoctoral Fellow, Saba worked on developing new tools to understand how cells use reactive redox signals as signaling molecules, and he developed a technique that they termed T-Rex, or targetable reactive electrophiles and oxidants, which is a chemistry-based tool that allowed for precise modification of redox sensor proteins. And since joining Randy's lab here at the University of Utah, Saba's continued to develop tools for understanding biological systems. And he has had funding both from the American Heart Association as a postdoctoral fellow, and more recently is funded by a K99 from the NHGRI, where Saba is working on the multiplexed interspersed CRISPR droplets, or MicDrop, which we'll hear much more about today. So with that, I will turn it over to Saba to tell you all more about his technique.
All right. Thank you so much, Zach, for the kind introduction. And thank you, Renee, and everyone at Bioprotocol Webinars for this invitation. I am delighted to be here to tell you a little bit about the technology that we have developed, MicDrop, that allows us to do large-scale CRISPR screen in an in vivo model system. So here's the outline for my talk today. I will begin by talking about why there was the need to develop this technology in the first place, go into a little more detail about how MicDrop works, and also during this process, answer some questions as well as considerations that one should take into account when designing a MicDrop screen. And near the end, I will give you some examples or snapshots of some larger scale MicDrop screens that we have performed so far, and end it all with after that, answering some questions from the audience. So let's get started. So I'm sure many of you in the audience are quite familiar with how revolutionary CRISPR-Cas9 technology has been, especially in terms of large-scale CRISPR genetic screens, they have become a powerful tool to be able to identify gene functions. However, most large-scale CRISPR-based genetic screens so far, they have been done in relatively simpler cellular model systems. And while these are incredibly powerful, they are phenotypically limited. You are limited to screening for only a handful of cellular phenotypes, such as looking at cell death, cell proliferation, changes in cell morphology, gene expression, and so on. Whole animal models, on the other hand, they display a range of complex phenotype, like animal development to various diseases such as cancer and metastasis, to various neurodevelopment and neuropsychiatric disorders, many of which are impossible to mimic or model in these relatively simple cellular model system. And therefore, being able to do large-scale CRISPR screen in these whole animal models will really open the door to study a lot of interesting and complex biology. And in this regard, and I think zebrafish are actually quite an ideal model system. And this is because they occupy a special niche in terms of their relatively high physiological complexity while still being amenable for high throughput genetic screens. And this is due to a variety of reasons, some of which include the fact that a single adult pair of zebrafish can lay hundreds of embryos, and you do need these embryos if you are planning to do any sort of larger scale genetic screens. Their fertilization is also external. This allows us to readily manipulate the genetics of these developing embryos, and their development is highly rapid. It takes only around 24 hours to go from a single cell organism to almost a fully formed one with many of its organs intact. So this allows us to do large-scale genetic screens in a relatively short duration of time. They are, of course, highly genetically tractable. In fact, 70 to 80% of known human genes have zebrafish orthologs, and they also display a range of complex morphological and behavioral phenotypes. So all of these factors combined make zebrafish an ideal model system for larger scale genetic screens. And my postdoctoral lab was in fact the first to show that CRISPR-Cas9 technology can successfully be used in zebrafish for genome editing. However, the current method, the traditional approach to make homozygous for phenotyping, is quite a long and laborious process. So it starts off with injection of CRISPR-Cas9 guide RNA complex in single-cell embryos, and then followed by multiple rounds of in-crossing and out-crossing to generate the homozygous, loss-of-function homozygous mutants. So as you can see, clearly this process is quite cumbersome to scale up. Not only do you have to generate these mutants separately, but you also have to keep track of all of these intermediates, which requires a lot of time, effort, labor, space, as well as cost, of course. So a better method would have been if you were able to screen for loss of function phenotypes directly in the F0 injected embryos, right? So this will eliminate the time required for keeping track of all the intermediates. And in this regard, there have been several developments including from our lab and several other labs, where people have shown that using multiplex guide RNA, so using guide RNA, more than one guide RNA targeting each gene, you can successfully recapitulate many known germline mutant phenotypes, like the ones shown here, directly in the F0 injected embryos. So this is great because this significantly shortens the time that's required for any large-scale genetic screen. But despite this development, you still run into this issue of, let's say, if you want to target 96 different genes, you first of all design the guide RNA library targeting these genes of interest. You mix it with Cas9 protein to make the ribonucleotide protein or RNP complex. But when it comes to injection, you are limited to basically targeting one gene at a time. So what you typically do in traditional F0 CRISPR screens is you take a microinjection needle, you load RNP complex targeting gene 1, let's say, and then calibrate this needle and inject a calibrated volume in a batch of embryos. And then if you want to target gene 2, you basically repeat this process all over again, this time taking a separate microinjection needle, loading it with RNP complex, targeting that separate gene, again calibrating it and injecting a calibrated volume in a separate batch of embryos. So you get the idea. If you want to target 96 gene or 500 genes, you just repeat this process all over and over again till you inject the number of genes that you want to inject. So as you can see, clearly there is no economy of scale. And again, when it comes to phenotyping, you basically keep these embryos in separate plates, grow them up to the desired time point, screen for phenotype, and then once you have identified a mutant that gives you the desired phenotype, basically correlate the phenotype with the genotype. So while this is a significant advancement over what the traditional approach of having to make germline mutants, for doing larger scale genetic screens, this method is not really amenable for doing scalable large scale genetic screens. So we ask, can we make this process even more efficient and develop a truly scalable platform? And that's where MicDrop comes in. So MicDrop stands for Multiplex Intermix CRISPR Droplet, and it truly is one of the first scalable platforms that allows us to do large-scale CRISPR screen in any in vivo model system. So how does MicDrop work? So the first step of MicDrop is generation of this CRISPR guide RNA library targeting the genes that you want to target. So this is identical to the step that I showed you before for the traditional F0 screen. We then, with these guide RNA, we add Cas9 protein to make the RNP complex. But unlike the traditional F0 screen, in MicDrop screen, what you do is add a unique DNA barcode that's associated with each gene. And I will come back to why this DNA barcoding is important. We then use a microfluidics device to generate nanoliter-sized droplets targeting these nanoliter-sized droplets that encapsulate the RNP complex targeting these various genes. And what these droplets allow us to do is intermix droplets targeting several different genes all together, loaded in a single injection needle, and then inject one droplet per embryo. We can then grow these injected embryos all together in mass, and then screen for phenotype at a desired time point. And then any mutants that display a phenotype that you are studying, you can isolate that mutant, recover the barcode from it, and sequence the barcode, which allows us to rapidly identify the causative genotype behind the phenotype that we observe. So this significantly streamlines the process of doing large-scale CRISPR screen in a zebrafish model system. So in the next couple of slides, I would like to go through each of these steps and get into a little more detail about how we perform each of these individual steps. So the first step of MicDrop is generation of guide RNA library targeting the genes of interest. So far, we have primarily employed this software called ChopChop to design our guide RNA. But of course, there are several other softwares that are widely available and widely used, such as CRISPR and CRISPR Scan, just to name a few. And users can use any of those software as well. We primarily use ChopChop because we think it's quite user-friendly, and also I like the graphical interface that they have. So all you need to do is basically input your gene ID or unsample ID for the gene that you want to target, specify the model organism for which you want to design your guide RNA against. And because we are doing CRISPR-Cas9 for knockout, you just specify that. And with the click of a button, it generates a list of guide RNA and arranges them according to a certain algorithm, prioritize certain guide RNA. So this is what a sample output looks like. So here we are looking at output for a guide RNA for targeting the RX3 gene in zebrafish. And as you can see, I really like this graphical interface because it shows you where these guide RNA, where in different exons do these guide RNA target. And it also uses a color code. So in green are guide RNA targets that are desirable, in yellow are less desirable ones, and in red are the least desirable guide RNA. And this desirability comes from the algorithm that the software uses, where it looks at various different factors, including predicted on-target gene editing efficiency, as well as off-target effect of each of these individual guide RNA. So now that you have a list of these guide RNA, we basically use a certain set of filtering criteria to select the most optimal guide RNAs for our screen. So typically what we do is we pick guide RNA that target early exons, so somewhere in the beginning 50% of the coding sequence of the gene. And this is just to allow us to mutate or knock out the gene in its entirety. We also prioritize guide RNA that have high on-target efficiency, and this is because so that we can see a high biallelic mutation rates in our screen. And also we prioritize guide RNA that have no predicted off targets, and this is for the obvious reason is that you don't want to confound the phenotype that you're observing because it is a result of off-target activity of the guide RNA rather than on-target effect. And a few other things, including the fact that we choose guide RNA that starts with GN so that we can do in vitro transcription with sp6-based polymerases. So now, after using these filtering criteria, we designed around 4 guide RNA targeting per gene. So some of the questions that we get asked often is how many guide RNA is optimal for targeting each gene. And so far we have mainly, for the screens that we have done in the lab, we have mainly used up to 4 guide RNA targeting each gene. And this is primarily so that we can increase the chances of seeing biallelic mutation. So even because we are not testing the efficiency of each of these guide RNA ahead of time, we are using 4 guide RNA so that there is redundancy, so that even if 1 or 2 guide RNAs were to fail, you still have a few guide RNA that will enable high gene knockout efficiency. But of course, this is something that you want to, there is a balance, the number of guide RNAs that you use, the chances of increasing off-target effect also increases, so you have to cut a balance in between high on-target activity, but reduced off-target activity. And another question that we often get, and this is particularly relevant to zebrafish as a model system, is zebrafish, because they have undergone an additional whole genome duplication effect, around 30% of zebrafish genes also have additional paralogs or analogs. So people often ask, should one target both analogs in the same animal? And my response to that usually is, it very much depends on what sort of question that you are trying to answer. So let's say if you are just looking at effect of knocking out a gene, you can target both analogs in the same animal, and this allows us to account for example, genetic compensation if you're only targeting one or the other analog. But let's say if you are trying to look at the effect of one particular one particular analog of the gene in regulating zebrafish developments, then it makes sense to target a single analog at a time. So the next step of MicDrop is generation of this droplet library targeting the genes that you want to target. So in our case, we actually repurpose a device that's traditionally used for digital droplet PCR to make these nanoliter-sized droplets. So this is what these droplets look like. They are highly uniform, around 100 micron in size, and each of these individual droplets, they carry the RNP complex targeting the gene that you want to target. So as you can see, these droplets are also slightly pink in color, and this is because we add a phenol red dye so that we can track the injection of these droplets in zebrafish embryos. And the great thing about these droplets is then you can intermix droplets targeting several different genes and then load it into a single injection needle to perform a micro injection in zebrafish. So here is a micro injection needle that's been loaded with droplets targeting, well, in this case, droplets that's been differently colored that I'm using as proxies for droplets targeting different genes. So you can see that you can pack these droplets quite tightly in these micro injection needle without having to worry about these droplets fusing with each other. We can now begin to inject these droplets in zebrafish embryos. So that's the next step of micro. So here is a video of me performing such an injection in zebrafish embryos. So for those of you who are unfamiliar with looking at zebrafish embryos, this is what it looks like. So to your left is this is the single cell, and this large blob over here is the yolk sac. So what you do is you inject one droplet in the single cell embryo, and then you push out the oil in between two adjacent droplets, and then inject the next droplet in the subsequent embryo. And you can continue injecting more and more embryos to target all the genes that you want to target. One thing we were pleasantly surprised to find out is that these droplets are actually quite stable. So you can store them at 4 degrees for an extended period of time, and they still retain their activity. So shown here, you can see that we see a very high phenotypic penetrance, so gene knockout efficiency, even after storing these droplets at 4 degrees for almost a month at 4 degrees. So this is great because this, this allows us to, you don't have the need to generate these droplets right before injection. You can generate these droplets ahead of time. And also this allows us to inject the same pool of droplets over an extended period of time. And this also in the future hopefully opens the possibility that we can generate these droplet libraries targeting hopefully genome-wide library and then share it with the wider zebrafish community. So one question we regularly get asked is how difficult is the injection and can it be automated? So all I'm going to tell you is that there is a slight learning curve to injecting, to how to inject these droplets, but we have successfully had graduate students as well as postdocs who have never had any experience with micro injection in zebrafish, learn these techniques just with one demonstration and be able to take this mic drop technique and become proficient at injecting these droplets. So I think anyone can use this technique to inject zebrafish embryos. And the question regarding can it be automated, this is something we haven't explored at the moment, but definitely something we are thinking about exploring in the near future. But again, I think the injection part surprisingly is not the rate limiting step. Zach here and other people in the lab, we have been able to inject more than 1,000 embryos in one sitting, so that hasn't been the bottleneck so far. So even automation is not going to really give us increase the efficiency any further. The next step in mic drop is, of course, the phenotyping step. So one question that we regularly get asked is what phenotypes can one screen for? So that's a great question. And I like to think that you can technically screen for any phenotypes that you can observe in zebrafish. So of course, the obvious morphological phenotypes, like the ones shown here, you can screen for any of those. For example, eye development, heart development, or trunk development, and several other and any other phenotypes that you can think of. Some phenotypic screen may require a little more user expertise depending on, or specific transgenic lines, depending on what is the phenotype that you are interested in looking for. in addition to that, of course, you can also look at various other behavioral phenotypes as well. And some of these projects are currently ongoing in the lab. And I will come back to a little bit to discuss a little bit more about this phenotyping, in zebrafish. And the final step of microdrop is, of course, recovering the barcode and then sequencing the barcode to find out what gene you mutated in each of these individual embryos. So in our case, we actually use a short double-stranded DNA around 137 base pair in length. And this part of this, we just end modify the DNA template that we use for in vitro transcription, and the rationale behind that is that, the spacer sequence for the guide RNA that we are using is unique to the gene that we are targeting. And therefore, by reading out the spacer sequence, you should be able to identify what is the gene that you are mutating. So we were pleased to see that co-injection of these DNA barcode along with the RNP complex doesn't cause any issues with viability or doesn't cause any significant toxicity in the injected embryos. One thing we were quite pleasantly surprised to find out is that these double-stranded DNA, as far as we know, they don't get integrated into the genome, but despite that, they are actually quite stable. So you can retrieve or PCR amplify these DNA barcodes even when injected at low concentration, even after 7 days post-injection. So this is great because this allows us to screen for phenotype that may appear at later stages of development. So most morphological phenotypes we can screen for at 3 to 4 days, by 3 to 4 days post-fertilization, but other interesting morphological and behavioral phenotypes, they start appearing at later stages of development. So the barcode being stable for, up to 7 days or longer really gives us a good handle to be able to screen these later phenotypes. So the other consideration that worth thinking about when performing large-scale microdrop-based screen is, in case of droplet injection, how many droplets to inject per gene? And another related question is what sort of phenotypes one can screen for, right? So these are sort of related, so that's why I put them together. And I briefly touched on this, what sort of phenotypes one can screen for. So again, depends on, I would like to think that you can screen for most observable phenotypes in zebrafish, but I think the phenotypes that you are going to screen for that are going to make the most likely target for genetic screens depends on how obvious is the phenotype, right? And also how distinct the phenotype is from non-specific phenotypes commonly observed in injected embryos. You don't want phenotypes that are very similar to non-specific phenotypes that you often see when you inject embryos, because then you will have a very high false positive rates. And also your phenotype, how many droplets you inject depends on how variable your phenotype is. If you have high variability from organism to organism, you will have to inject a larger number of droplets. But if your phenotype is quite consistent, you can get by injecting only a few embryos per gene. So now that I have talked about each of these individual steps and all of the things that one should think about when planning a microdrop screen, let's look at some of the genetic screens that we have done in our lab. So one example application that I would like to share with you is we use microdrop to see if we can use this technology to identify novel regulators of cardiovascular development in zebrafish. So I use existing RNA-seq datasets to identify a list of around 500 different genes that are enriched in zebrafish embryonic heart. And we hypothesized that these genes are likely important for heart development. And when we cross-correlated these list of genes to what is known about them, we found out that indeed the genes shown here in orange dots, indeed, a knockout of these genes already have reported phenotypes and already have reported phenotypes in ZFIN that knockout of these genes result in disruption in cardiovascular development in zebrafish. So we were quite confident that we are indeed enriching for genes that are important for heart development in zebrafish. So I then use a filtering criteria to remove any duplicate or unannotated genes and genes with known phenotypes. I was left with around 200 different genes about which not much was known. I then designed guide RNA targeting these genes. And like I mentioned to you before, we prioritize guide RNA with no predicted off targets, and around 80% of our guide RNA targeting the genes, these 200 or so genes, had no predicted off target. Around 20% of the genes had off targets with 3 base pair mismatches. So this, again, based on certain studies, these, even though they have off targets, these guide RNA should not be very efficient at targeting at the off target region. I then generated the droplets targeting these 200 or so genes, and then injected them in zebrafish embryos. So this is what the results look like. So compared to uninjected droplet injected embryos, we were pleased to see that there was no significant change in the viability. So this is great because you don't want the droplet injection itself to have any associated toxicity. But this also tells you that the genes that we are targeting don't have early lethality phenotype. And then I grew these embryos and then screened for heart developmental phenotypes at different embryonic stages. So at 24, 48, and 72 hours for post-fertilization, by which most of heart development in zebrafish is complete. So as expected, these are, we identify a handful of embryos where we see just gross morphological defect. And this is expected. You do see that whenever you are performing any sort of injection in zebrafish embryos. To us, what was most important, we see around 25% of the injected embryos that have cardiovascular phenotypes. And these are the embryos that I was interested in. So I isolated these embryos, retrieved the barcode from them, and sequenced the barcode to find out what gene knockout resulted in the phenotype that we observe. So from this screen, I was able to identify, from the screen of these 200 or so genes, I was able to identify around 10 candidate hits, mutation in which result in cardiovascular development in zebrafish. So I won't have the time today to go through each of these individual mutant phenotypes, but I just want to share with you a few here. So one mutant that we identified is in a gene called CLEC19A. Again, a poorly characterized gene. Not very much is known about this gene. And what we found out is that mutation in this gene caused an obvious edema in the mutant. So you can see, compared to wild type, you can see there is a bulging of the pericardial sac. So if you look closely at this zebrafish heart, and that's one of the beautiful things about being able to work with zebrafish, is that they're highly amenable for imaging because they are transparent at these early stages. So you can see this is a wild type heart. So unlike mammalian heart, zebrafish heart is two-chambered with an atrium and a ventricle. And you can see, compared to wild type, the mutant heart is of course linear, and also there is an extension of the atrioventricular canal. We can also look at the beating pattern of this heart. So this is a wild type heart. You can see smooth and rhythmic beating pattern of the atrium and the ventricle. And this is the mutant to the right. So you can see not only the morphology is quite distinct from the wild type, also, if you look closely, you will also see some blood flowing back into the atrium from the ventricle. And this is likely due to malformation of the atrioventricular canal. The other mutant that I'm going to share with you today is a mutation in a specific isoform of glutathione transferase called GSTM3. So mutation in this gene resulted in ventricular arrhythmia. And we were able to do some cardiac conduction or voltage mapping experiment in collaboration with Callum McRae's lab and show that indeed they have significant delay in cardiac conduction. The mutant hearts has significant delay in cardiac conduction, as well as there is significant reduction in median action potential duration, which is likely manifesting into the ventricular arrhythmia that we observe. And there was a recent paper that showed that GST-M3 is indeed a risk factor for a disease called Brugada syndrome, where patients harboring this mutation have arrhythmia, also show arrhythmia. So the genes that we're identifying are also disease relevant as well. And the final mutant that I'm going to share with you today is not really a heart phenotype, but it is a blood phenotype. So you can see mutation in this gene result in extreme porphyria. So the mutants completely lack the development of red blood cells compared to the wild type over here. And you can inject this, you can rescue this phenotype as well as some of the other phenotypes I mentioned to you previously by co-injecting the guide RNA along with mRNA expressing the protein. So this is just to show that the phenotype that we are observing is a result of on-target activity of the guide RNA rather than off-target effect. And one of the reasons I wanted to share with you this mutant, besides the phenotype being quite striking, is the fact that we were not looking for a blood phenotype. We were looking for a heart phenotype, but we were able to identify a blood phenotype in our screen. So you can see that mic drop can not only be used to identify the genes responsible for a particular phenotype, but can also be used to identify novel phenotypes that are associated with different genes. So aside from using mic drop as a functional genetics tool, we were also curious to see if mic drop can be used as a target ID platform. So for those of you who are familiar with chemical biology a little bit, one of the big challenges in chemical biology is to identify the gene targets of various small molecules and drugs. And we think mic drop is quite well suited for these kind of screens. So we asked, can we use mic drop to do a mock screen to identify the gene targets of this small molecule called optivin? So this small molecule, optivin, was identified in our lab as a small molecule that causes light-dependent motion response. So if you take zebrafish larvae and shine light on them, not much happens. But now, if you pre-treat these larvae with optivin and then shine light on these samples, you see this very vigorous light-dependent motion response. And we know from our lab's work is the gene target of optivin is an ion channel protein called TRPA1B. So if you were to knock out this gene, you completely ablate this phenotype. And the beauty of zebrafish is that they are so tiny that you can array them in 96-well plates and then look at their motion response in a high throughput format. So shown here is zebrafish embryos that have been arrayed in 96-well plate. And you can see each of these red lines are tracking their movement patterns. So you can see you only see movement when you treat these embryos, pre-treat these embryos with optivin, and you completely lose this phenotype if you use guide RNA to target the TRPA1B channel. You can also quantitate this phenotype in a high throughput format. So shown here is movement in response to 5 separate pulses of light. You can see, again, you only see response when you treat the embryos with optivin before shining light on the sample. And you ablate this phenotype again if you previously inject guide RNA targeting the TRPA1B ion channel in these injected embryos to knock out the TRPA1B gene. So we asked, can we perform a mock screen to retrospectively identify TRPA1B as the gene target of optivin? So we did a mock screen where we combined droplets targeting the TRPA1B gene with non-targeting scrambled droplets in a 20:1 ratio, loaded them in microinjection needle, and then injected 1 droplet per embryo. These embryos were then grown up to 4 or 5 days post-fertilization, and then I arrayed them in 96-well plate, treated them with optivin, and then asked which of these embryos don't show any light-dependent motion response. So this is what a representative data looks like. So you can see, as expected, most of these embryos show light-dependent motion response. There are a handful of embryos, like the ones highlighted here, where you see either a reduced movement or no movement at all. So I isolated these embryos, retrieved the barcode from them, and sequenced to find out what the genotype of these embryos were. And in 100% of these cases, we're able to find out that the gene mutated in these embryo is trait A1B. So you can see that we can use MicDrop as a target ID platform as well. So in summary, what I have shown you is that we have developed this technique called MicDrop that allows us to do targeted, systematic, large-scale, and truly scalable large-scale genetic perturbation in a whole animal model system. And I have shown you some examples demonstrating that MicDrop is indeed a robust platform for large-scale CRISPR screen in zebrafish model system. And we can use this screen, combine that with various morphological and behavioral phenotyping approaches downstream. I also showed you an example showing you that MicDrop can be used as a target ID platform as well. And even though I showed you a relatively smaller scale screen targeting only around 200 or so genes, we are quite confident that this technology can be scaled up to target hundreds, several hundreds, if not thousands of different genes. And in fact, Zach and several other people in the lab have been working, doing much larger scale screen targeting 700 to 1,000 different genes. And more excitingly, we also have ongoing discussions and collaborations to perhaps generate genome-wide, CRISPR droplet library or MicDrop library that can be shared with the broader zebrafish community. So we are really excited about this technology, and we are looking into expanding the applications of this technology further. So a few things that I'm really excited about is being able to deliver other CRISPR-Cas9 system using this MicDrop approach. So one thing I want you to take away from this is that MicDrop, in essence, is actually quite a powerful delivery platform. You can basically deliver any cargo using these droplets, and depending on what cargo you are delivering, you can do different sort of screens. So for example, if you deliver CRISPR-Cas13D, you can do gene knockout instead of gene knockdown. You can do gene knockdown instead of gene knockout using CRISPR-Cas9. You can also perform CRISPR inactivation or activation screen. You can perhaps deliver morpholinos, or you can deliver mRNA to do gain of function screen rather than doing loss of function using CRISPR-Cas9. But aside from targeting just the coding genes, which account for less than 2% of the entirety of the genome, we are also looking into the possibility of targeting various interesting non-coding genomic region. We have these vast 98% of the genome that has been relatively unexplored. But now with this MicDrop technology, we have the ability to systematically knock out these interesting non-coding genomic regions to be able to look at their functions in regulating animal development and behavior. And finally, one of the things that we're really interested in is basically able to remove the phenotyping bottleneck. So I showed you how, using MicDrop technology, how we have been able to successfully remove the injection bottleneck in zebrafish embryos, so the screening bottleneck. But one of the things that have become now challenging is being able to do the phenotyping at large scale. So if we can remove the phenotyping bottleneck, I think that will be really really outstanding, because then we will be able to truly do much larger scale genetic screen. So one of the things that we think about is basically combining looking at not only morphological and behavioral phenotypes, but also looking at various cellular phenotypes using state-of-the-art techniques such as single-cell RNA sequencing. So being able to combine the large-scale CRISPR screen using MicDrop with various other downstream phenotyping approaches will really open the door to look at a lot of interesting and complex biology. So with that, I would like to thank various members of the Peterson Lab who have who have helped with the development of this platform and taken this platform and really applied it to answer a lot of interesting and complex biological questions, and Zach being one of them. I'm also thankful to all of our collaborators who collaborate on various aspects of this technology, and I'm also grateful to all of our various funding agencies that has funded this work. And last but not the least, thank you all for joining, and I would love to take any questions.
So, thanks, Saba. I tried to answer a few of the questions that were put into the Q&A if there were maybe more straight methodological questions. So I've answered a few of those, but I will pose some of these other ones to you. How does the method account for cell and tissue specificity, or maybe does it? Would be another way of asking that.
Yeah, I think that's a great question. At this moment, we are doing knockout. We are not doing tissue specific knockout. We are doing knockout in the entire organism. But you can possibly imagine being able to do tissue-specific knockout if you were to express, let's say, the Cas9 enzyme in specific tissues using a tissue-specific promoter. And all you need to do is then inject the guide RNA, and you can hopefully see tissue-specific knockout in that case. But for now, all of the screens that we have done so far have been by just doing knockout in the entire organism.
All right.
And one thing, Zach, you have worked on this. You worked using this method as much as I have done, and probably more. So please chime in if you have additional either things to add or any clarifications to make regarding any of the questions.
Sure. Could you elaborate more on, I guess, how you detect whether the guide RNA is generating an indel in an injection, and is that a part of the screen or not?
Yeah, that's a great question. So right now, if you are doing a large-scale genetic screen, it is not very convenient to, first of all, test the guide RNA beforehand if these guide RNA are most optimal in generating indels. So what we have done is we have validated at least the ones with hits that you are indeed seeing the after you do a screen, once you identify a hit, validate that those guide RNA have high on-target activity. We do validate those. And what we typically do is basically amplify the genomic region where you anticipate the mutation to happen, and then use next generation sequencing to find to see that there is mutation. You can also just use basically some sort of T7 endonuclease assay, or there are other techniques, to be able to look at whether you are seeing high mutation efficiency or not. But to be able to systematically do for each and every individual gene, I think is going to be costly, and also is going to be difficult to do. But of course, if you already have validated guide RNAs that work well in zebrafish, you can always use those guide RNA and know that they will give you high editing efficiency.
Somebody noted that since you're injecting some of the oil outside of the cell and you lose some of the droplet, you're not injecting the entire droplet. So how much does that matter? Sort of how much of an entire droplet needs to be injected for that to be a good injection, I guess?
Yeah, that's a great question. Actually, that's already factored into, that wastage of a little bit of droplet in the beginning and at the end. We already take that into account when making these droplets, so the concentration of Cas9 and the RNA inside these droplets is actually high, so that even if you end up injecting only 50% of the droplet, you should still see pretty high editing efficiency in the injected embryos. Yeah, that's a great question. And most of the time we end up injecting anything from 50 to 75% of the droplets and wasting a little bit in the beginning and at the end.
Thank you. This may be a quick answer. Do you know if there's any pre-designed guide RNA libraries that are commercially available, I guess specifically for zebrafish in this question? But I think the answer more globally is probably yes, but I'm not sure about zebrafish.
Yeah, I think you're right, Zach. I think more globally, it's yes, people have done these larger scale CRISPR screen primarily in cellular system, and have shown or looked at target efficiencies, validated target efficiency for each of these guide RNA. For zebrafish, there are smaller screens that people have done and reported efficiencies of these guide RNAs. So some work from Sean Burgess' lab and other labs as well, where they have validated guides, but not at genome-wide scale, so you will have to look up case-by-case basis. I think ZFin has a good collection of these guide RNA, but not exactly, it doesn't really tell you whether these guide RNA are efficient. You will have to look at these individual papers that have used these guide RNA.
Yeah. I'm going to sort of combine two questions here. Fatma in the chat had expressed some confusion on how to know whether a specific gene was causing a specific phenotype, and then Delia was asking in terms of doing those complementation experiments to show the specificity where you're injecting that mRNA along with or with the guide RNA. Adelia's question was, when do you inject that mRNA? Are you doing it together with the guide RNAs in the F0? Since that would be, timing would be pretty critical for a lethal gene, for instance.
Yeah, that's a great question. So yes, one way we show that the phenotype that we are observing is a result of on-target effect rather than off-target activity is by rescuing that phenotype by expressing the gene that you are targeting, right? So and that's done using co-injection of the mRNA along with the guide RNA. And you're right that the timing of it is going to be critical. But so far, what we have done is basically co-injected the guide RNA and the mRNA together, and then looked at whether the phenotype gets rescued, right? But you can always do the I don't know, we have never tried it, but it is possible that maybe you can control when the mRNA gets expressed, or when the gene gets expressed by using, plasmid injection or different tune or using promoters that are regulated, let's say by heat shock promoter or something, so you can use those to regulate when the mRNA gets expressed. But in our case, because we are directly injecting mRNA, this gets expressed as soon as it gets injected, and most of the phenotypes that we have looked at are early developmental phenotypes. So, if we see a rescue using this method, we get reassured that the phenotype we are seeing is a result of on-target activity.
Okay. There's two different questions asking for maybe some further elaboration or clarification on barcoding and how it works. So Brajesh had asked, how do you use the barcodes to tag them to the guide RNA? And could you explain, another person asked if you could explain again how the barcode works.
Slide. All right. So what we are doing, again, I think this is just one way of doing the barcoding. You can make your barcodes in many different ways. All you need for barcode is basically, as long as you know what gene you are targeting, and there is a unique sequence that's associated with it that you can read out and know, and that will tell you what gene you mutated in that particular embryo, right? So that is the whole point of this barcode, because when you're injecting droplet, even though I have shown it in colors here, these are all pink in color, so you have no way of knowing what gene you are targeting in each of these individual embryos. And what barcode allows you to do is that rapidly identify what is the what is the mutation in each of these individual embryos, right? So what the barcoding technique that we have done, that we have used in our lab, and this is simply because it's the most convenient one for us, so what we have done is basically to generate the guide RNA, you have to first make a DNA template, right? To do the in vitro transcription to make the guide RNA. And this usually has the sp6 promoter and as well as your spacer sequence, because this is what specifies where the guide RNA targets, right? But what we have done is to make the DNA barcode, we basically amplify that sequence and end modify it. And this end modification is done so that the barcode has higher stability. So these barcodes are modified with biotin at the 5' end. But we have also shown later that actually you don't require these modifications. You can simply just amplify your DNA template for in vitro transcription and then use that as the barcode. So what you do is you make your RNP complex, add this short double-stranded DNA in the RNP complex, and then make the droplets, and then mix the droplets, inject the droplets. And when you are injecting your RNP complex, you are also injecting this unique barcode in each of these individual embryos. And after you find a mutant with a phenotype, what you do is basically use a set of primers at the T7 and T7 termini, using that binds to these regions, and amplify this DNA barcode, and then Sanger sequence it to read this spacer sequence that tells you what gene you mutated in that particular embryo. So hope that is clearer than what I explained previously.
I think so. Maybe anyone who is that could chime in a chat and let us know.
But like I said, you can use any plasmid DNA or any double-stranded DNA with a unique sequence as long as you know that one gene corresponds to one sequence.
Yeah. Another question that had come up, I think both on the Q&A on the website page, but also once or twice here in the Q&A, was a lot of questions about use in other species, and not even just other animals, but could it be used in plants or, C. elegans, nematode? Can you maybe just comment on feasibility of adapting it to other species, and if there's specific things you would have to do for that?
Yeah, I think those are great questions. I'm not familiar with how gene editing is done in plants, so I can't really comment very much on that. But in terms of being able to translate it to other model systems, such as Xenopus, or where you do embryo injection, it's definitely feasible. So we are looking into expanding this technology and be able to do CRISPR screen in Xenopus, for example, because those are most similar to zebrafish in terms of the size of the embryos, as well as the number of embryos that you get from mating adult organisms, right? So that will be the most obvious model organism of choice to translate this technology. Regarding C. elegans, again, I think from what I know a little bit, I think the injection is slightly more challenging. You do have to inject it into the gonads of the organism, so that becomes the more rate limiting step rather than doing the injection, rather than packing these or targeting several genes at a time, so. But again, I haven't worked with C. elegans very much, so I don't exactly know. But if you think you would like to try out this mic drop technology in C. elegans, feel free to reach out to us, and we can certainly help you out with what things will need to be modified for you to be able to translate this technology in C. elegans. Did I miss anything, Zach, or did I answer?
No, I don't think so. I think, it comes down to, obviously, if you're wanting to use it specifically for the guide or for CRISPR, you'll have to change organisms for designing that. But then I think, yeah, if it's an animal that can undergo injection, then it should be relatively smooth, and the things that you would have to change will be, volumes and concentrations based on what's worked in those before. Yeah.
Exactly. Yeah. Thanks.
Gwen had a question wondering, would like to know why you choose guide RNAs that are cutting only in exons versus including ones that might cut at the transcriptional start site. And maybe you can just comment more broadly on, choosing where to target them.
Yeah, I think that's a great question. And I think especially with, recent discussions regarding genetic compensation, that has been a thing that we have been thinking about a lot, where what is the ideal place to target these guide RNAs at. So far, most of the screens that we have done, we have targeted, early parts of exon, and this comes primarily from the fact that we have seen high knockout efficiency and high phenotypic penetrance for genes with known phenotype. We have been able to recapitulate several known mutant phenotypes by targeting just this early coding exons. But that can be an alternative strategy if you are trying to target transcriptional start site. That's something we haven't validated, but that remains to be seen if we can get better efficiency and perhaps eliminate genetic compensation by targeting the transcriptional start site, for example. One thing we have done is in certain cases when the gene is small, we have tried to target, tile these guide RNAs so we can take out majority, if not the entirety of the gene. That is another strategy that we have used so that we can minimize genetic compensation. So again, for the transcriptional start site, we haven't validated, but that's a strategy that can be used. And if you're willing to try it, please try it and let us know how it works, because it will be simply changing the guide RNA design criteria, and the rest of the steps are going to stay the same. So it'll be pretty easy to accommodate.
Awesome. I think that's the majority of the questions that
okay.
see.
Yeah, I think if you have further questions, if you put down in the, I think webinar link, I don't know if that will still stay active, but I can go through it later and answer some of those questions as well. If you have questions, all feel free to reach out to us through email, and we will be happy to assist you if you are planning to use this technology either to do CRISPR screen in zebrafish or try it out in other model systems as well. And we have been trying to advertise this technology, and several people have reached out to us, and we have been at least trying to be as helpful as possible by showing other people how to do it, either flying them here and show them how to do these injection, or going to their labs and doing a workshop. If there is a lot of interest, we can even think about doing a workshop at specific universities, to demonstrate these technologies. Yeah, so we can look into that. So do reach out to us and let us know if we can be of any help.
All right. Well, I think that's a great time to end. Thank you so much for this interesting talk, Saba and Zach, and also for great moderation. And thank you everyone for the lively discussion. As Saba already mentioned, we do have a Q&A section at our website, so you can visit that and still post questions or participate in a discussion if you have an answer. And as I also mentioned earlier, the webinar is being recorded and will be posted on the website later. That might take a couple of weeks, but please come back if you want to watch it again or have specific questions. Again, thank you so much for coming today, and I hope to see you next time. Bye, everyone.
Recording stop.